So what is AlphaFold? The short answer is that it's basically like DALL-E, the image generating AI, except for proteins instead of images. Instead of putting in a string of text as your prompt to get a picture, you put in a string of amino acids to get a 3D protein structure. So why is that useful? Well, effectively it means that biologists can now take any sequence that they find and get a really good approximation of what the structure will probably look like. And this is incredibly valuable because a lot of biology involves figuring out how proteins interact with each other and what structural features of a protein are responsible for its function. By understanding the structure, we can start to understand how certain elements affect the function of the protein. And we can figure out the amino acids of a protein from its genetic sequence, and that's really fast with today's tools. So from the genetic sequence alone, we can approximate the amino acid sequence and then thus the protein structure that comes from the gene. This capability is far beyond anything we had 20 or even 10 years ago, to be honest. I mean, the hypotheses that we can generate now are, well, they're just a lot faster than in the past. Like, for instance, during my masters, I used AlphaFold to predict how a disordered bit of a protein would form into an ordered section when it interacts with another protein. And it actually correctly modeled this in such a way that I was able to cross-reference with some data from the paper that I read from that I hadn't actually seen yet. And it was correct. And it was so useful for my hypotheses. Okay, so what is AlphaFold3 then compared to AlphaFold2? So from my understanding, AlphaFold3 can now model post-translational modifications and interactions with certain small molecules, such as DNA. And this is important because post-translational modifications can massively affect a protein structure, and they're crucial for protein interactions, especially in eukaryotic biology. AlphaFold3 now accommodates these modifications, which were actually missed in AlphaFold2. Okay, so then what are the caveats? Even though AlphaFold provides a hypothesized structure, it is literally just that, is a hypothesis. So while AlphaFold is incredibly accurate compared to previous methods, the structures it generates are literally, they're not proven. And so until you validate the structure yourself with techniques like X-ray crystallography or cryo-EM, you can't be certain that it's actually correct. And it would be bad science, basically, to base further work on what is essentially just a guess. Ideally, AlphaFold should be used alongside structural techniques like, X-ray crystallography or cryo-EM, that sort of stuff. I'm excited for the future of AlphaFold, actually, for versions 4, 5, 6, 7, and beyond. I hope that future iterations are going to include molecular dynamics, because currently AlphaFold doesn't actually have a physics model. I mean, you can technically argue that it had to learn physics to some extent to model the structures, but that's a philosophical debate. It doesn't have a simulation that simulates physics. Future versions with molecular dynamics would be amazing, because current molecular dynamics techniques are really slow, even on modern super quick graphics cards. Simulating a single protein folding in place is slow already, and adding interactions with other proteins or even the whole environment slows it down so much further. Simulating an entire cell is just completely out of our reach at the moment in our lifetimes. But if something like AlphaFold comes along for molecular dynamics, we might one day simulate an entire cell. This would be a huge breakthrough for medical applications, for synthetic biology, generative AI for creating new biological pathways, and making value-added bioproducts. There's so many things that would come out of molecular dynamics from AlphaFold, and I'm really hoping that one day we get there. If you like this video, please let me know, and I will probably make more. I really like doing this. It gave me a reason to get outside and to take my pet for a walk. And yeah, I don't know. It was fun.